So please understand, in my last session, I just spoke about something which is called as TS of Anther. Let us first of all once again revise that particular thing and then move ahead. As I have told you, the outermost covering is called as epidermis. It is a single layered cells, flat cells, single layered, barabar. Inner to that there is presence of something which is called as special type of single layer of cells. What those cells are called as? Endothelium. Please keep on drawing along with me so that it doesn't become lagging. I told you that the cells of endothelium, their radial walls and transverse walls are thickened. Their radial walls and transverse walls are thick. Have I told you this? They show a characteristic thickening. This characteristic thickening is called as callus. What is called as? Yes. The callus is hygroscopic in nature. What it is? It is hygroscopic in nature. Okay. It helps in dehiscence. Barabar. I told you in the groove there are some cells which are not thickened, and these are the cells which will form the line of dehiscence. Barabar. Inner to that there will be presence of cells, smaller cells, about 2 to 3 layers of cells which will be there. Middle layer cells. Right? Inner to that, what starts is something what is called as the pollen sac starts over here, and inside that pollen sacs there are cells which are large enough, pyramidal in nature, okay, placed at intervals. Barabar. These are called as cells of tapetum. Barabar. So this is your first layer, that is called as epidermis. This is the second layer, that is called as endothelium. Okay. Cells of the middle layer. Barabar. And the innermost layer, that is called as tapetum. Barabar. Inner to that there will be microspore mother cells which will undergo meiosis and will keep on producing microspores. So these are microspores. I'm just giving you an idea in this, the sectional view of right. Have you finished? Okay. Will you please write? In short, we will write, since you don't have anything in front of you, that's why. Otherwise, I would have made you read that particular thing. Correct? Please write TS of Anther. TS of Anther. The TS of Anther shows two layers. The TS of Anther shows two important parts. A. Anther wall. Anther wall, and B. Microsporangia or pollen sacs. Anther wall and microsporangia or pollen sac. First point, क्या आएगा? Anther wall. The anther wall consists of following parts. The anther wall consists of following parts. A. What is that? Epidermis. Okay. Single layer, flat cells, protective. It consists of single layer of flat cells, they are protective in nature. Single layer of flat cells, protective in nature. Second, endothelium. एक लाइन छोड़ के लिखना if possible. Endothelium. How many layers? Tell me. One common layer for all. One common layer for all microsporangia. A single common layer for all microsporangia. What is the specificity? Their walls show characteristic fibrous thickening. Walls show characteristic fibrous thickening. Walls show characteristic fibrous thickening. Called callus. Called callus. Barabar. Next point, it is hygroscopic. It is hygroscopic. It is hygroscopic. And what it helps in? Dehiscence of anther. It is hygroscopic. Helps in dehiscence of anther. Dehiscence का मतलब breaking open so that pollen grains can be released. So first point क्या लिखा? Single layer common to all microsporangia. Second, shows characteristic fibrous thickening which is called as callus. Barabar, it is hygroscopic. Fourth point likha, it helps in dehiscence of anther. And likha, idhar tak ho gaya sabka likh ke. Fifth point, the cells of the endothelium in the cells of endothelium present in the groove between the two anther lobes. In the groove between the two anther lobes are unthickened and they form the line of dehiscence. they form the line of dehiscence. The cells of endothelium which lie between the groove or in the groove between the two anther lobes are unthickened, they are not thick, and they form the line of dehiscence. What they form? Line of dehiscence. I hope you understand. ठीक है? समझा कि नहीं समझा? Did you get my point properly? Next, what is the next one? Middle layer. Third one, middle layer. Okay, it consists of 2 to 3 layers of cells. Parenchymatous cells ही होते हैं ये भी. It consists of 2 to 3 layers of cells. Next point, they degenerate at maturity. They degenerate at maturity. Next, tiptum. Barabar? How these cells are? Are they small or little bigger in size? Bigger in size. What is the structure? Pyramidal, na? Okay. Okay. Pyramidal cells located at intervals. Pyramidal cells located at intervals. I am making it short so that you can. Pyramidal cells located at intervals within the microsporangia or near the microsporangia. Right? How their ploidy can be? They can be diploid, triploid or polyploid. Please write. They can be diploid, triploid and polyploid. Next point. They are nutritive in nature. They are nutritive in nature. They secrete UBISH, UBISCH, UBISH granules. UBISH granules. They secrete UBISH granules for maturation of microspore mother cells. And cause microsporogenesis. They secrete sporopollenin. Which forms the exine of what? It forms the exine of microspores or pollen grains. Exine of microspores या pollen grains. Understood? Perfectly? ठीक है? Last B. A था A was anther wall. Now we go to B part. What is B part? Microsporangia. What you'll write? क्या लिखोगे? What you'll write? It is also called as pollen sac. Please write. Also called as pollen sac. It bears microspore mother cells. It bears microspore mother cells. At maturity, next point, at maturity, these undergo meiosis. To form what? To form microspores. Or pollen grains. Okay. Now we'll look for the process of microsporogenesis. Okay. माइक्रोस्पोरोजेनेसिस कैसे होता है ये हम लोग अभी देखेंगे. जरा यहां पे देखिए प्लीज. You know that any part of the plant actually develops from meristematic tissue. Barabar? The meristematic tissue gets a stimulus to convert into a young pollen, a young stamen. Barabar? In the young stamen, the meristematic tissue distinguishes into, okay, primary parietal tissue, primary parietal tissue, which eventually develops into anther wall. Okay, and the other is primary sporogenous tissue. The primary sporogenous tissue undergoes mitosis, and this replicates time and again to produce specific type of cells called as archaeosporial cells. क्या कहते हैं हम लोग इनको? Archaeosporial cells. The archaeosporial cells may themselves act as microspore mother cells, or they will undergo further mitosis and eventually lead to formation of microspore mother cells. Please remember, microspore mother cells are diploid in nature. What they are? Diploid. So if I take a microspore mother cell which is diploid in nature, this is my microspore mother cell. At maturity, the microspore mother cells undergoes meiosis. You know, I have taught you meiosis. There will be first division and there will be once again division, and will eventually form 4 microspores, barabar, haploid microspores. But when the microspore mother cell undergoes meiosis, it tends to produce 4 pollen grains, okay, to form a tetrahedral tetron of microspores या pollen grains. बराबर? So there is 1, 2, 3 and the 4th one is on the back side. That is why you will see a tetrahedral tetron. This microspores will eventually separate and form unicellular uninucleate structure, which is basically called as pollen grains or microspores. So all these are microspores or pollen grains. Please remember, these microspores are haploid in nature. What they are? So in a young stamen, the meristematic tissue undergoes a stimulation to form a young stamen. In the young stamen, as you know, filament is there and anther is there. Especially in the anther, okay, the anther is developed by two different types of cells. Barabar. The meristematic tissue forms primary parietal tissue and primary sporogenous tissue. The primary parietal tissue develops into anther wall. आपको पता है ना, parietal layer and visceral layer. Parietal मतलब बाहर का, visceral मतलब अंदर का. So बाहर का is going to form the anther wall, while the primary sporogenous tissue undergoes mitosis to form archaeosporial cells. These archaeosporial cells may directly act as microspore mother cells or they undergo mitosis. So equational division होगा and it will produce microspore mother cells. Once the microspore mother cells are formed, each microspore mother cell, each of the microspore mother cells under the influence of tapetal cells will undergo a process called as microsporogenesis. Where each microspore mother cell will undergo meiosis to form 4 microspores. But these microspores are not directly formed in a separate manner. They form a tetrahedral tetron, which is basically like this, of microspores. As they gradually mature, each of which gets separated to form a single unicellular uninucleate structure called as pollen grain. This formation of unicellular uninucleate structure of pollen grain is called as microsporogenesis. क्या कहते हैं हम लोग इसको? I hope it is clear. Clear? Please write this followed by that. What happened? From here you are supposed to continue over here. You can write ना microspore mother cell, वही लिखा है ना इधर. So इसके नीचे यही लिखो, आपको समझ में आएगा. Here it is. The word archaeosporial cell is very crucial. Huh? Please remember, you will see such cells not only in development of male gametophyte, but you will also see it in female gametophyte also. Right? Same word, archaeosporial cells. Not understood. We have almost learnt everything of this in Kingdom Plantae when I taught you angiosperms. Barabar, but that was in a little brief. This is in very much detail now, because this is sexual reproduction in flowering plants. The flowering plants दो ही type तरह के होते हैं, angiosperms and gymnosperms, and that too predominantly angiosperms is what we take into consideration. That's why. So तभी भी वो tetrahedral tetron था, तभी वो माइक्रो, बट ये जैसे डिटेल्स थे, जैसे archaeosporial cells and those things were missing because that was not part of our syllabus itself. That's why. This year you will see everything much in detail when this specific topic. I guess everyone has written it. Barabar. Now there are famous questions which come on this particular aspect. Barabar. I'll just give you 4-5 before that we'll go and complete development of male gametophyte first of all, and then we'll go to the 5 different types of numerical questions which they might ask you. 5 forms are there, 5 types are there potentially. Okay. So can I wipe it off? Okay. Let us first of all understand the structure of, so this is called as microsporogenesis, right? Okay, you will be able to write it, right? In the form of note if it is asked, you will be able to write it, yes or no? Yes. Okay. We go further. Please give the title development of, structure of pollen grain. Structure of pollen grain. Structure of pollen grain, please look over here. A typical pollen grain in case of angiosperm is a unicellular uninucleate structure. It shows presence of single haploid nucleus and it shows presence of cytoplasm. Mostly the cytoplasm is granular in nature. Barabar. It has got its own plasma membrane, but it is externally protected by something which is called as sporoderm. What is called as sporoderm? The sporoderm consists of 2 layers. The outer layer is called as exine. What it is called as? While the inner layer is called as intine. What it is called as? The size of this microspore, like this is microspore. If this is the microspore, it is about 25 to 30 microns. Please remember the size. They can ask you the question on this. What is the size? Okay, 25 to 30 micron in size. Barabar. It has got 2 external covering. The outer covering is called as exine. The exine is made up of a chemical called as sporopollenin. This is naturally produced. This is naturally produced, and it is the one which cannot be degraded by any enzyme. It is resistant to changes in temperature. It is resistant to acids. It is resistant to alkali. So you can say that it is the world's naturally produced most resistant chemical, and that is why some of the pollen grains can retain their capacity to sprout and produce male gametophyte for years after years. You can keep the microspore stored, preserved for long time, because they are covered externally by something what is called as the sporopollenin. I hope you understand. But this sporopollenin, which is exine, barabar, it is not completely going to cover it, barabar. There are certain areas which are unthickened areas, barabar, and these unthickened areas where either the sporopollen is missing or it has been thinned out, this particular area is called as the germ pore. क्या कहते हैं हम लोग इसको? So this is called as the germ pore. What is called as the germ pore? Okay, the inner lining the inner lining is called as intine. It is made up of pectin and cellulose. Pectin and cellulose. बराबर? So क्या क्या अभी तक मैंने बताया? A typical microspore is unicellular, uninucleate. It has got a haploid nucleus which is surrounded by cytoplasm. Okay, it is about 25 to 30 micron in size. बराबर? It is externally covered by a protective covering which is called as sporoderm. क्या कहते हैं हम लोग इसको? The sporoderm has got two layers, the outer layer and an inner layer. The outer layer is called as exine, while the inner layer is called as intine. The exine is made up of sporopollenin, while the intine is made up of cellulose and pectin. Barabar? The exine at multiple areas, okay, is unthickened, and these unthickened areas are called as germ pore. In case of dicotyledonous plants, there are more than one germ pore, so it is multiporate, while in case of monocots it is always going to be uniporate, that is single germ pore is going to be there. Okay, so this is all regarding what? The microspore structure. ठीक है, please write one by one. फटाफट लिखो. First point. What happened? Anything you have not understood? Yes, please. क्या? Structure of microspore or pollen grain. Okay. First point. It is unicellular, uninucleate. It is a unicellular, uninucleate structure. Formed by microsporogenesis. Formed in microsporogenesis. It shows haploid nucleus surrounded by cytoplasm. It shows haploid nucleus surrounded by cytoplasm. Haploid nucleus surrounded by cytoplasm. Third point, it is about 25 to 30 micron in size. It is about what? 25 to 30 micron in size. Okay. It is externally covered by sporoderm. It is externally covered by sporoderm. Externally covered by sporoderm. Okay. The sporoderm consists of two layers. The sporoderm consists of two layers. What are the two layers? A, exine. Exine. It is made up of sporopollenin. It is made up of sporopollenin, बराबर, sporopollenin, बराबर ना. Sporopollen is कैसा है, क्या बोला था मैंने, what I have told you, it is okay, it cannot be acted upon by any of the enzymes. Second मैंने क्या बोला था, it is going to be acid resistant, alkali resistance, okay, okay, right. Sporopollen is hardest naturally occurring substance. Sporopollenin is hardest naturally occurring substance. Sporopollenin is hardest naturally occurring substance. Resistant to acids, alkalis, and also to temperature and pressure changes. Also to pressure and temperature changes, बराबर. Hence microspores or pollen grains can be, hence microspores or pollen grains can remain intact for long duration. In time. It is smooth, delicate, made up of pectin and cellulose. It is smooth, delicate, made up of pectin and cellulose. Okay? Is it okay? Next. What is next? Okay. At certain areas, at certain intervals, such okay, the exine is either absent or unthickened. Okay, such areas are called as germ pores. Such areas are called as germ pores, either absent or unthickened. Such areas are called as, how many germ pores in monocot? One. That is why uniporate, and dicots may many, that is why multiporate. Okay. Since okay, monocots, pollen grains of monocots show a single germ pore, they are referred as uniporate. While in dicots they show many germ pores, hence multiporate. Correct? Did you get my point? depending upon the type of pollination, please write, depending upon the type of pollination, the external surface of, depending upon the type of pollination, the external surface of pollen grain shows variability. What it shows? Variability. For example, for anemophily, ANEMOPHILY, in bracket you write wind pollination, the external surface is smooth. The external surface is smooth. Okay? While for entamophily, entamophily is with the help of insect pollination. So it is for entamophily, insect pollination. How it is? It is sticky and spiny. It is sticky and spiny. समझा कि नहीं समझा? Okay. Now I'll just tell you one important aspect. देखो सुनो मेरा बात, ये एनसीईआरटी का बहुत इंपॉर्टेंट पॉइंट है, इसके ऊपर बहुत बार क्वेश्चन आता है. ये जो यंग पोलन्स होते हैं, ओके, इन यंग पोलन्स के वजह से प्रॉब्लम्स आते हैं, और कुछ अच्छा भी है कुछ बुरा भी है. देयर आर टू थिंग्स विच यू नीड टू रिमेंबर. इफ देयर आर सर्टेन इंडिविजुअल्स, हमारे में कुछ इंडिविजुअल्स ऐसे हैं, if the pollen grains go into their nasal tract, see 25 to 30 micron is very tiny, okay. There are some human individuals, if certain pollen grains of certain plants they go into our nasal tract via respiration, okay, they cause inflammation of the upper respiratory tract, okay, which we call it as hay fever. It's a kind of allergy. What is called as hay fever or allergy. So they can cause allergies, barabar. But some of the, especially pollens, okay, are consumed by athletes and horses also. Some of them are fed to the horses also. Why? Because they have a tendency, okay, that they increase the amount of RBCs in your blood, and that is why your oxygen carrying capacity increases, so you can have lot of oxygen while running at a very fast pace. So ऐसे pollen products बनाए जाते हैं, बराबर. There are certain pollen products which are made, okay, and fed to especially athletes and especially horses, okay, who are runners. But there are some individuals who are, if they respire such pollen grains, then they may suffer the inflammation in the upper respiratory tract, which is called as hay fever. What it is called as? It's a kind of allergy. These people are very sensitive. They develop something called as hypersensitivity. Hypersensitivity का मतलब है कि वो एक पर्टिकुलर इंडिविजुअल के लिए सेंसिटिव रहेगा, लेकिन दूसरे इंडिविजुअल के लिए वो सेंसिटिव नहीं रहेगा. फॉर मी इट मे नॉट बी वेरी मच सेंसिटिव, बट फॉर यू अदर्स यू मे हैव द प्रॉब्लम. उनको स्नीजिंग चालू होता है, कफ चालू होता है, ओके, इमीडिएटली एवरीथिंग स्टार्ट्स बिकॉज द बॉडी वांट्स टू डायलेट एवरीथिंग आउट. That is why sneezing, uncontrolled sneezing, 10-12 sneezing आते रहते हैं. If they respire, upper respiratory tract पूरा inflamed हो जाता है, okay, bronchospasm आ सकता है अगर और ज़्यादा बढ़ गया तो. So such individuals should relocate from that area. Otherwise they'll keep on entering into respiratory episodes which may take their life also. Okay, so that is called as hay fever or pollen allergies. क्या कहते हैं हम लोग इनको? Okay. समझ में आ गया? This is what is most commonly seen with respect to problems of pollen grains. Correct? तो नीचे लिख के रखो. pollens in some human individuals may develop, okay, hay fever or pollen allergies. You had asked me something. What happened? What is the doubt? फूलसूंगा तो एकदम एक्सट्रीम हो जाएगा. हाँ, but ये usually happens in with the pollens when they are given out, no, and when they are traveling via air, you tend to respire them and still you suffer from it. सूंगा तो मर ही जाएगा. It will be heavier, very very heavy allergy. क्या बोला मैंने? Pollen in some individuals, okay, may develop. In some individuals may develop hay fever or pollen allergy. Okay, pollen allergy resulting in inflammation of upper respiratory tract, URTI, inflammation of upper respiratory tract, next, pollens are also used as a supplement to athletes and race horses. Athletes and race horses. Especially the bee pollens. Especially the bee pollens, which is known to enhance the performance right? ठीक है? Close the books and complete concentration over here. Whatever I am telling you, please understand it. So we have learnt up to this point something called as structure of microspore. Barabar? Now let us see how the development of male gametophyte takes place. See, pollen grain itself is a partially developed male gametophyte. Partially developed. In NCERT they simply say that it is male gametophyte. But actual male gametophyte ka development kaise ho raha, that we need to understand. See, when the pollen is formed, it is formed in the form of a unicellular uninucleate structure which is going to show covering. This is called as the exine. And you know, at certain intervals there is presence of germ pores, right? Yes or no? Now what happens is about 60% of the pollen grains, see, when the pollen grain is matured, barabar, it undergoes first mitotic division. And the first mitotic division results in formation of a two-cell structure. How many cells? Two cells. A small cell with a larger nucleus, a small cell with a larger nucleus, and a large cell with a smaller nucleus. A small cell with a larger nucleus and a large cell with a smaller nucleus. The one which is with smaller size but large nucleus is called as the generative cell. Barabar? While the one with large cytoplasm but small nucleus is called as the vegetative cell, or it is also referred as tube cell. Okay, you will still have the covering will be still there. That covering is sporoderm ka covering, barabar, with germ pores at irregular intervals. So about 60% of the pollen grains are shed at this point only. क्या बोला मैंने? You developed a single cell, barabar. It has undergone first mitotic division to form two cells. What are the two cells? There is a tube cell with a small nucleus and a one with the large nucleus is called as. Now देखो tube cell जो होता है, in tube cell, okay, there is proper cell wall, उसको cell wall है. There is presence of proper, इसके अंदर आप अगर ध्यान से देखोगे, you will see that there will be presence of vacuoles. There will be presence of starch granules and there will be presence of a small nucleus. ये तीनों चारों चीजें यहाँ पे नहीं होते हैं. There is no cell wall in this particular area because majority of the area is covered by the nucleus itself. There is no vacuole, no chance, no proper cell wall, no vacuole, okay, no starch granules, and we have a large nucleus. So this is the difference between a tube cell and a vegetative cell. Now once this happens, about 60% of the pollen grains are released in this stage only, barabar, while the remaining ones, okay, are shed afterwards, barabar. What is that? Once this first meiotic division happens, there will be a second meiotic division, and the second meiotic division leads to formation of, okay, the generative cell also divides to form two more, the generative cell divides to form two more nuclei. I hope you understand. And these are called as male gametes. What they are called as? So before this happens, this particular pollen grain is going to get shed. So once it sheds, it goes to the, this is your stigma. Once it goes to the stigma in a two-cell stage, barabar, the stigma identifies whether the pollen grain is of its own type or not. There is an interaction which is called as pollen pistil interaction. What it is called as? Pollen pistil interaction. Okay, the surface of the pollen grain has got specific chemicals which allows the stigma to understand whether this pollen grain is of its own type or of any other type, whether the pollen grain is of the same species of the plant or any other species of the plant. So stigma is capable of identification whether the pollen grain belongs to its own type. If it belongs to its own type, actually it secretes moisture as well as a little bit of a very important chemical, mucilaginous chemical is going to come out. As a result, the pressure inside this particular cell, especially the tube cell, will increase. And once it increases, okay, from one of the area where there is potential area for what? If there is a germ pore, then the intine pops out. The intine pops out, barabar, along with the pollen tube, along with the tube nucleus, barabar, and the cytoplasm. The cytoplasm starts oozing into the pollen tube, barabar. Now, once the cytoplasm starts oozing out into the pollen tube, before it becomes completely emptied, the generative cell undergoes division. The generative cell undergoes mitotic division to form two more cells, barabar. So first mitotic division is followed by second mitotic division and you will get two male gametes. So now you will have, this is your pollen grain, it has produced a pollen tube. I hope you understand, barabar. It shows presence of tube nucleus at the tip, barabar. It shows presence of the cytoplasm which is present all across, and there will be formation of two haploid non-motile male gametes, barabar, which is formed by the division of the generative cell. I hope you understand. The generative cell divides to form two haploid non-motile male gametes, and this structure which you get, that is the pollen tube, barabar, plus tube nucleus plus two haploid non-motile. Please remember, अभी तक जितने भी आपने plants में पढ़े, majority of them were motile male gametes. These are non-motile male gametes. Along with the cytoplasm, that is all together referred as male gametophyte. What is called as male gametophyte. Okay? जो आप बड़ा देखते हो root stem leaf वाला, वो होता है sporophyte. और ये है आपका मेल गैमेटोफाइट, अ प्लांट विच इज प्रोड्यूस मेल गैमेट्स. हम कहते हैं प्लांट और अ पार्ट ऑफ द प्लांट विच प्रोड्यूसेस स्पोर्स इज कॉल्ड एज स्पोरोफाइट. सो दिस इज अ प्लांट ऑफ द पार्ट और अ पार्ट ऑफ द प्लांट विच इज प्रोड्यूस व्हाट, मेल गैमेट्स, एंड दैट इज व्हाई इट्स कॉल्ड एज मेल गैमेटोफाइट. आई होप यू अंडरस्टैंड. That is why microspore is referred as male gametophyte, because it is the one which produces male gametes. समझा? Did you get my point? So how many mitotic divisions have happened? 2. Barabar na? The first mitotic division results in formation of tube cell and generative cell, while the second mitotic division happens only in generative cell, not in tube cell. Barabar? And thus results in formation of an elongated structure. Okay? आप अभी याद रखो मेरा एक बात, बहुत इंपॉर्टेंट बात बोल रहा हूं, यह बाद में भी दिखेगा आपको. You have seen hibiscus plant, बराबर? Hibiscus plant produces this, बराबर, pollen tube सब produce होता है. For the elongation of the pollen tube, a specific type of mineral called as boron is important. बराबर? अगर ये boron नहीं है आपके soil में, तो elongation of the pollen tube will not happen. In Maharashtra, the soil is very, very poor in boron. And that is why the elongation of the pollen tube does not take place, and that is why throughout the year you will see presence of a lot of flowers getting produced on hibiscus, but there is neither the seed nor the fruit. किसी ने देखा है hibiscus का fruit? किसी ने देखा है hibiscus का seed देखा है कभी? No. हम लोग hibiscus को propagate करते हैं by cutting. हम लोग cut करते हैं और उसको लगा देते हैं. So by cutting we propagate that particular thing. So that is why you must remember this aspect. What is that? Development of male gametophyte. क्या क्या बताया देखो. The microspore or the pollen grain is a unicellular uninucleate structure. Just before it undergoes pollination, okay, it divides to form, okay, it divides, the single nucleus divides by mitosis to form two cells, okay. A large cell with a small nucleus called as tube cell or vegetative cell, and a small cell with a large nucleus called as generative cell. What? Generative cell, right? Okay, we have seen that particular thing. Once majority or like 60% of the pollen grains are going to get pollinated in how much? 2 cell stage, barabar. As they reach the stigma identifies whether the pollen grain belongs to its own type. If it belongs to its own type, then it secretes specific type of germinating agents. It allows the pollen grains to germinate. Okay, it secretes moisture, as a result of which the pressure, the cytoplasmic pressure inside the tube cell increases, and the intine comes out from one of the germ pore, forming a pollen tube, the nucleus and the tube nucleus and the cytoplasm all enter into the pollen tube and extend the pollen tube. Just before the entire cytoplasm enters the pollen tube, the generative cell divides and forms, okay, two non-motile haploid male gametes which enter into the pollen tube. Thus, a pollen tube will have totally 3 nuclei. The apical one will be the tube nucleus, while the 2 latter ones will be the. समझ में आ गया? Did you understand? Thus, a pollen tube which contains tube nucleus, 2 haploid non-motile male gametes along with the cytoplasm is referred as. Yes. So this is called as development of male gametophyte. I hope you have understood. हाँ के ना? Sure समझ में आया? Yes or no? Okay. Do you want this to be written? I want you to draw the diagrams. I'll just draw the diagrams once again and you draw it. I'll draw it first of all. Look over here once again. Draw with me if possible. What we have? We have a unicellular uninucleate structure. What is this structure? Microspore. What it has? The germ pores, right? Are yes or no? Right? It undergoes, this is before pollination, before it gets moved from one flower to the other flower, it moves from anther to the stigma. Before that, that is, this is before pollination, it undergoes first mitotic division, barabar. This first mitotic division will lead to formation of two cells, right? Correct? The upper cell is called as vegetative cell, or it is called as what? Tube cell. While the second is called as what? Generative cell. Barabar? In this stage, 60% are going to get, they will get pollinated. Barabar? So this is two cell stage. As they get liberated onto the stigma, so after deposition on stigma, appropriate stigma, that too, barabar ki nahi? What happens? One of them, it develops what? Pollen tube, right? Are हाँ के ना? Correct? The generative nucleus will be over here and the cytoplasm must be pouring inside. Yes or no? ठीक है ना? So what this is? This is pollen tube, right? And the last, what will happen? You will see that the pollen tube has elongated. Yes or no? You will see the tube nucleus towards the tip, barabar. You will see cytoplasm, yes or no? The entire cytoplasm has now shifted inside the pollen tube, correct? And you are having this generative nucleus or generative cell has got divided into not one but two haploid non-motile male gametes. So this is pollen tube, this is tube nucleus, this is cytoplasm, and these are two haploid non-motile male gametes. So this is your development of male gametophyte. What this is called as development of. Now I'll write 5 different mathematical questions. You will say, sir, biology में किधर मैथ्स आता है? तो इधर आता है मैथ्स. Okay, drawn. Please write 5 important questions. So, do you remember why 2 male gametes? Anyone remembers? When the sexual reproduction takes place in case of angiosperms, the first male gamete fuses with the female gamete, while the second male gamete fuses with the secondary nucleus, forming the primary endosperm nucleus, which ultimately develops into endosperm. I hope you remember that. Barabar, that is why there is formation of 2 haploid non-motile male gametes. So should I give the questions now? Okay. First question. Question number 1. How many meiotic and mitotic divisions are required for formation of? How many meiotic and mitotic divisions are required for formation of? How many meiotic and mitotic divisions are required for formation of 1000 male gametophyte? If the question is asked, how many meiotic divisions and mitotic divisions are required for formation of 1000 male gametophyte, how you will deal with the question? Look over here. I have got 1 microspore mother cell. Correct? Or it is also called as PMC, pollen mother cell. This is diploid in nature. Yes or no? It undergoes division to form 4. I hope you understand. It undergoes division. Single meiosis produces 4. So, 1 PMC से, okay, 1 PMC leads to formation of 4 microspores, barabar, and each of the microspore will develop male gametophyte. Yes or no? So अगर 1 से 4 आता है तो अपने को कितने चाहिए? 1000, बराबर. तो 1000 से डिवाइड करो किसको? 4 को, तो आपको कितना आएगा? So we require 250 meiotic divisions. What we require? 250 बार meiotic division हो गया तो अपने को 1000 माइक्रोस्पोर्स मिलेंगे. 1000, उस 1000 में एक बार meiotic division मतलब 1000 बार meiotic division होगा तो आपको 1000 वेजिटेटिव सेल और जनरेटिव सेल मिलेंगे, बराबर. और हर जनरेटिव सेल में वापस एक बार माइटोटिक डिवीजन होगा तो अपने को एक मेल गैमेटोफाइट मतलब 2000. आई होप यू अंडरस्टैंड. तो एक मेल गैमेटोफाइट मिलेगा, बराबर. मतलब दो माइटोटिक डिविजन रिक्वायर्ड है तो 1000 मल्टीप्लाई बाय 2. So that is how many? 2000. I hope you understand. So how many meiotic divisions? 250. And how many mitotic divisions? Please write. ऐसा लिखो पहले. One microspore mother, one pollen mother cell divides by meiosis to form 4 pollen grains. Therefore, for 1000 male gametophyte, 1000 divided by 4, 1000 divided by 4, that is 250 microspores, 250 meiotic divisions, 250 meiotic divisions barabar, which will give 1000 pollen grains or microspores, barabar na. Next, each microspore undergoes 2 mitotic divisions for formation of male gametophyte. Each microspore undergoes 2 mitotic divisions barabar for formation of male gametophyte. Hence, 1000 into 2, 1 or 1000 into 2, that is 2000 mitotic divisions are required. A slight change in the question. Okay? See, you need to be very, very accurate with respect to what has been asked. If they ask you 1000 male gametes, how you will deal? If they ask you 1000 male gametes, how many meiotic and how many mitotic divisions are required for formation of 1000 male gametes? अगर ये क्वेश्चन है तो कैसे करोगे आप? I don't know. One male gametophyte contains how many male gametes? That means potentially they have asked you a question. How much? 500 male gametophyte कैसे करेंगे? I hope you understand. नहीं समझा? Did you get my point? आपके एक मेल गैमेटोफाइट में कितने मेल गैमेट्स होते हैं? 2. आपको कितने चाहिए? 1000 मेल गैमेट्स. मतलब आपको कितने मेल गैमेटोफाइट चाहिए? Did you understand the difference between gametophyte and gametes? This is a gametophyte and these are, okay, in one gametophyte there are 2 gametes. Therefore, in 1000 male gametes, okay, will be 500 male gametophyte. तो अभी आपको पता है, ओके, समझा ना? मियोटिक डिविजन्स के लिए क्या करने का? डिवाइड बाय 4. सो 500 डिवाइड बाय 4 कितना आता है? 125. और अगर आप कितने करोगे? आप इंटू 2 करना पड़ेगा आपको. 1000 माइटोटिक डिविजन. सो आंसर क्या आएगा? 125 मियोटिक डिविजन्स एंड 1000. प्लीज राइट. पूरा लिखने का जरूरत नहीं, अभी तुमको समझा. So you just write 1000 male gametes are present in 500 male gametophyte. समझा कि नहीं? नेक्स्ट, How many meiotic divisions are required? How many meiotic divisions are required? How many meiotic divisions are required for formation of 53 male gametophytes? 53 male gametophytes. Very easy it is. हाँ, डिवाइड बाय 4 कितना आता है? कितना आता है बताओ. हाँ, 53. You have to divide by 4, बराबर ना? So how many it will come to? Okay, so how many meiotic divisions? हाँ, 13 will lead to 52. For the third one, that is 53rd one, you have to perform one more. You cannot have half the meiotic division. ऐसा नहीं होता है. 0.125 नाम का कोई meiotic division नहीं है पॉसिबल. तो आपको कम से कम कितने करने पड़ेंगे? 40. तो 14 की वजह से कितने आएंगे? 56. बट 56 आ गए मतलब 53 आ गए ना. When you have 100 rupees, that means you also have 90 rupees with you. Yes or no? So always you have to go one more. I hope you understand. ऐसा अगर ऑड नंबर में क्वेश्चन आया तो दिया रहेगा ऑप्शन में 13.12, तो वो करेक्ट नहीं है. बिकॉज 13.12 करके meiotic डिवीजन होने वाला नहीं. इट विल बी 14 meiotic divisions which will lead to 56. that will be 53 also. Please understand this. समझा? ओके, हो गया. नेक्स्ट, ये सब पैटर्न्स है. See, every pattern is different. Okay, please write. A plant has produced 560 microspores. 560 microspores. Okay, 560 microspores. Okay, count the number of नहीं 560 नहीं 320 ले लो. 320 microspores. Count the number of microspore mother cells in each pollen sac. (320 माइक्रोस्पोर्स. अब ये कैसे करोगे? थ्री 320 पोलन ग्रेन्स है बराबर. तो 320 पोलन ग्रेन्स यू हैव टू डिवाइड बाय व्हाट? 4. कितना आ गया? 80 बराबर. बट एक एंथर, कितने एंथर लोब्स होते हैं? 4 एंथर लोब्स ना. तो 80 डिवाइड बाय, समझा कि नहीं? समझा? सी, आई हैव 320 pollen grains, right? 1 PMC produces 4 pollen grains, right? So 320 pollen grains will be produced by 80 pollen mother cells, right? But in a stamen there are 4 microsporangia, barabar. So 80 will be divided by 4. So which comes to 20. So 20, 20, 20 and 20. I hope you understand. Clear? So how many? 20. फोर्थ क्वेश्चन हो गया ना. अब फिफ्थ एंड द बेस्ट ऑफ ऑल. बराबर. Let us see who can do it, because this requires ultimate understanding of whatever I have taught. If you have understood, then you will be able to get me a correct answer. Okay, please write. Count the number of pollen grains. Count the number of pollen grains. Count the number of maximum pollen grains. Count the number of maximum pollen grains which can be produced by 10 flowers in a pea plant. 10 flowers in a pea plant having 4 PMCs, 4 pollen mother cells, in each microsporangia. This requires little bit of understanding from 11th standard ka portion also. Such type of question were usually asked in as like those questions which are very difficult to get. This is a level 3 question. Level 1, level 2, level 3. This is a level 3 question. This requires understanding of whatever you have learned as well as something extra also, which you have learned previously. What is the answer? How you did 640, tell me. How you did 640, you let me know, na? How many PMCs? 4 PMC. Barabar? So you multiplied it by what? 4, na? 4 क्या? 4 meiotic division, ना? 1 से 4 आएगा, इसके लिए कितना आ गया? 16. बराबर? उसके आगे 16 into 4 क्यों? 4 लोब्स है, इसके लिए 16 into 4, हाँ 64. बराबर. एंड कितना फ्लावर्स? 10 फ्लावर्स. बराबर ना? बराबर ना? 640 आता है ना? बराबर कि नहीं? ठीक है. अरे लेकिन पी प्लांट के अंदर देयर आर 10 स्टेमेंस. वो कौन मेरा चाचा दिखेगा? Pea plant क्यों दिया? Because it contains 9 plus 1 arrangement ना. So what was the part which was missing was the number of stamens. बराबर. So कैसे है देखो. सबसे पहले नंबर ऑफ पीएमसी. नंबर ऑफ पीएमसी कितने हैं? 4. बराबर. दे विल अंडरगो मियोसिस मतलब 4. सो ये कितना आ गया? 16. सो दिस इज गोइंग टू बी द नंबर ऑफ पीएमसी. बराबर. ऐसे जाते हैं. फ्लावर्स का नंबर कितना है? 10. बराबर. नंबर ऑफ स्टेमेन्स इन ईच फ्लावर कितना है? कितना है? 10. सो दिस बिकम्स 100. बराबर. अरे आके ना. देन नंबर ऑफ ये pollen sac, हर एक में 4, so it becomes 400, yes or no? बराबर. Then number of PMCs कितना है? 4, so that becomes 1600, बराबर कि नहीं? And then number of meiotic divisions कितना? 4, so कितना आया? So the missing part was what? So you missed the figure by just 0, isn't it? You missed the figure just by how much? 0. So you should remember everything. You should remember the examples also, because it's a famous example, Dithecus anther ka. Clear? I hope you understood this. समझ में आ गया? So we have finished up to development of male gametophyte. Clear? In our next lecture we will go for development of female gametophyte and double fertilization. I hope you have understood all these parts. Okay, from your NCERT का जो आपका 12th स्टैंडर्ड का बुक है, मैंने बोला तुम लोग को NCERT का 12th स्टैंडर्ड का बुक अभी तुम लोग पढ़ सकते हो up to development of male gametophyte, and you can solve from your MCQs. Next time I want the challenger का book over here, and you are supposed to solve the first. You have got a challenger ka book with you. That challenger book you bring, you solve all the MCQs up to development of male gametophyte. Okay? ठीक है? That's all. Thank you very much.